Hey y'all, so in this short lecture, what we're going to talk about is how RNA is processed. In other words, how do our cells take what's known as a precursor mRNA or a pre-mRNA and trim it in a way so that it becomes what we call the mature RNA, or sometimes we just say mRNA. So let's imagine that this is an RNA molecule that has just been transcribed. We're going to go ahead and label our 5' end and our 3' ends. And remember, the long line here represents our backbone, while these ones here represent our individual nucleotides of this RNA molecule. And RNA undergoes a couple of modifications after transcription in order to take it from what we call that pre-mRNA to the mature mRNA. And let's focus on two of those modifications right here. The first one is that the 5' end is modified with a guanine nucleotide. We call this the 5' cap. Oftentimes it's just drawn like a little circle like this one is here. And this guanine is actually put on, in some ways we kind of describe it as being put on upside down. And so if, for example, the mRNA sequence at the beginning at this 5' end here was AUG, UCA, that guanine sometimes would be written something like this here, upside down, to indicate that modified cap. Now on the 3' end, what we have inserted is a polyadenylation tail. And this poly A tail is generated by proteins after the RNA polymerase has passed the poly A signal, which we talked about as we were discussing transcription. With poly A tails, the A here stands for adenine, and so what's going to happen is these proteins are going to add on a string of adenines that are anywhere between 50 to 250 nucleotides long. Now, both the 5' and the 3' modifications that occur here are really important. In fact, both of them are required to help stabilize this mRNA and prevent it from degradation. In addition, the 5' cap and the poly A tail help to facilitate export from the nucleus. And finally, the third thing that they do is that they aid in the ribosome attaching to the mRNA. And so, in other words, they're going to aid in the process of translation. Now, in addition to the 5' cap and the poly A tail that's at the 3' end of an mRNA, we find additional regulatory elements in here. Before the start codon, which is going to be AUG, and we'll talk about codons in more detail when we talk about translation, there is a region known as the 5' untranslated region, or the 5' UTR. After the stop codon, which can be something like UAG, there is what we call the 3' untranslated region. These are part of the mature mRNA, and they may contain in them sequences that actually control how well this mRNA is going to be translated. If you're interested in this topic, what you might want to look up are what's known as microRNAs, or sometimes these are abbreviated as miRNAs. And these have really gained a lot of interest from researchers in the past 25 years as a way for controlling translation of proteins. But in addition to these 5' and 3' untranslated regions, the pre-mRNA may contain regions that we call introns and exons. Introns are intervening sequences. These are ones that are going to actually be excised or removed, and so they are not part of the mature mRNA. However, exons are the expressed regions of mRNA, or those that are going to be part of the mature mRNA at the end of RNA processing. So let's talk a little bit in detail about how the eukaryotic cells remove these introns and kind of paste back together these exons. So what I've done here in this image is I've labeled for you exons with an E and then introns with an I. Again, the exons are the regions of the mRNA that are going to be part of the mature mRNA. The introns are these intervening sequences. And so what the cell is actually going to end up doing is it's going to employ the use of enzymes and RNA, a complex known as the spliceosome. And the spliceosome is going to recognize sequences at these junctions of the intron and exon. It's going to cut the intron out, and it's going to paste back together these two ends of the exon. And so these spliceosomes actually have some what we call nuclease activity, which allows them to cut the DNA, and they also have ligase activity to paste the RNA back together. So then let's go ahead and take a look at what's happening in detail here. Essentially, what we're going to find is that these regions of exons, which I'm drawing here in purple, are going to be brought close together, and the intron is basically going to kind of be folded outward, like so. Spliceosomes are going to bind to these regions, or these junctions between the exon and the intron. They're going to make cuts at these two sites. And as those are cut, the spliceosome is going to paste back together those exons to form the mature mRNA. And I'm just going to draw a line right here to indicate where that cut occurred. And that intron sequence will actually be degraded at this point within the nucleus. One thing to point out is this whole process here of RNA processing occurs in the nucleus before the RNA is exported for translation. One other quick note that probably is of great importance is that this RNA processing only occurs in eukaryotes. In prokaryotes, there are no introns and exons. All of the mRNA that is transcribed is the mature mRNA. In addition, in prokaryotes, there are no 5' caps or poly A tails, and so the mRNA in prokaryotes is not protected in the same way as it is in eukaryotes. So just to summarize the big ideas here, in this process of RNA processing that we've talked about, we talked about two types of modifications that we see. The first included modifications to the 5' and the 3' end. These were the cap at the 5' end and the poly A tail at the 3' end. And we discussed how these are important for protecting the mRNA from degradation. They help in translation by allowing the ribosome to attach to the mRNA. And then they also facilitate the export of the mRNA from the nucleus. The other part we talked about were what's known as introns and exons, and how introns are cut out and exons are then spliced together to form the mature mRNA. One thing we did not talk about here, but you might want to look into, is how there is what we call alternative splicing, in which a single pre-mRNA might encode for 2 or 3 different versions of the same protein based on which sequences are identified as introns or exons.